Welcome back, guys. In this video we're going to see or start a new playlist for physiology. Yes, it's a very basic discussion, but without without knowing this, you should not go to exam hall. I hope you understand the importance of this topic. Yes, it's nothing but transportation across cell membrane. Okay, guys. Yes, before starting this video, I request you to please like this video and comment your feedback on my video. That's it, guys. If you are a new user, please go through the playlist, so you will find many useful high-yielding videos. Okay, now we can start. The transportation across the cell membrane. So what's meant by that? Yes, any ions or proteins which are transported across the cell membrane can occur in two mechanisms. One is active mechanism, where ATP has been used. The next is passive mechanism. Yes, first we can see the difference between these two, active mechanism and passive mechanism. Passive mechanism is like a downhill. You are going down the hill. Okay, in other words, in physiological term we can say it as it's along the electrochemical gradient. Yes, it's along electrochemical gradient. This is against electrochemical gradient or uphill. You are spending energy for the transportation. Yes, here no energy is required in passive, energy is required in active in the form of ATP. So what can be the example of passive diffusion? Simple diffusion. It can be simple diffusion, facilitated diffusion, the next non-ionic diffusion, the next is osmosis. In active diffusion, primary active transport, secondary active transport. The secondary active transport is further divided into symport, antiport. Yes, this is the overall classification of the transportation across cell membrane. Okay, now I will focus on passive diffusion. Yes, in passive diffusion, what can be the example for simple division? We can discuss simple division now. Yes, what comes under simple division? Simple division is nothing but the diffusion of gases occurring in our respiratory tract, that is through the alveoli, and also the lipid soluble substances like alcohol and steroids which has been taken up by the body. So all this will be coming under simple diffusion. In simple diffusion we have to study one formula according to Fick's law. Yes, Fick's law is nothing but the net transportation is equal to minus dA gradient of C and the gradient of X. So what is this? This D is nothing but diffusion coefficient. What is this A? A is nothing but surface area. And this is concentration gradient. The gradient X is the thickness of the membrane. So how to deal with this formula is, any type of diffusion, that is any type of transport, is always directly proportional to surface area. If the surface area increases, this net transport also increases. If the concentration gradient between the membrane increases, this net transport also increases. That is, the numerator are directly proportional. The denominator, that is thickness, is inversely proportional. That is, if the thickness increases, the net transport decreases. For example, I will give one example for this. Example, in emphysema, what can be the net transport? how the net transport can be influenced by this? In emphysema, obviously there will be a decrease in surface area. So this decrease in surface area decreases the net transport, or it decreases, in simple words, it decreases the diffusion. Okay, I will give another example. What if there is pulmonary fibrosis? Yes, in pulmonary fibrosis, the thickness of the membrane, respiratory membrane, is obviously increased, thereby decreasing the diffusion capability of the gases. I hope you understand. Always thickness is inversely proportional to the net transport. Yes, the next is facilitated diffusion. Facilitated diffusion occurs in the direction opposite to electrochemical gradient, but it never uses ATP. That's the difference. So what is uses for the transport? Yes, there will be a carrier media. Yes, this carrier media always follows two rules. It is always stereospecific. That is stereospecificity. The next rule is it follows saturation kinetics. Yes, I will explain this. Stereospecific. Take an example, GLUT transport. Yes, it is specific for glucose and galactose. It can't transport any other compounds other than this. So it is specific to glucose and galactose. So that's meant by stereospecific. What is meant by saturation kinetics? Saturation kinetics is nothing but this carrier protein, when gets saturated with the traveler protein, it can't further increase the rate of transport. That is, it comes to certain amount and it remains stable. Yes, if for example, if one GLUT can transport one glucose, for an example, we are having 50 GLUT and 100 glucose. This 50 GLUT can pair only one glucose each, so it can carry only 50 glucose. So from there it becomes saturated. It can carry only 50 glucose, so in that it gets saturated. That is saturation kinetics. I hope you understand. Even there are 100 glucose, there is only 50 carrier proteins. It can carry each one, so it becomes saturated at 50. That's the point. The next is non-ionic diffusion. The real-time example is secretion of NH3 in kidney. NH3 secretion occurs through simple diffusion in PCT, whereas in collecting duct, it occurs in the form of non-ionic diffusion, and this NH3 combines with H plus and forms ammonia and get excreted in urine in collecting duct. The next is osmosis. This is the last heading from passive diffusion. In osmosis, there is a transfer of molecules from lower concentration area to the higher concentration area through the semi-permeable membrane. Yes, we all know from 5th standard, that's osmosis. It comes under passive diffusion. The next, we are going to start active diffusion. Yes, uphill transport. Here we are using ATP as an energy gradient. So you have to travel or make the molecule or ion to travel across the cell membrane against the electrochemical gradient in this. So it can follow primary active transport or secondary active transport. Secondary active transport is of two types. One is import, another one is antiport. In primary active transport, we can see an example of sodium potassium ATPase. This is sodium potassium ATPase pump. Yes, this is the pump. It plays an important role in transporting across the cell membrane in each and every cells in our human body. Yes, it expels out 3 sodium ions. This is the first step. The second step is it gives two potassium ions into the cell. Yes, there is a net loss in this. The net loss plays an important role in maintaining the cell volume. Yes. The next is secondary active transport. In secondary active transport, we can take this as symport. We can take this as antiport. Yes, in symport, we can take this cell as the interstitial cell of PCT. When there is an electrochemical gradient created here after the activation of sodium potassium ATPase, this electrochemical gradient can be overcome by the symport of sodium glucose or also sodium amino acid. Yes, this is symport. The other example is sodium inorganic phosphorus. Sodium iodine symport that occurs in thyroid. This is PCT, etc. Whereas in antiport, let us take this as the cell of myocardium. Yes, in myocardial cell, this electrochemical gradient is overcome with the antiport mechanism, thereby making the calcium inside the cell to go out. So we can give a living example in this. So what can be an example? Digitalis inhibits this sodium potassium ATPase pump, but if it get inhibited, this also will get inhibited. There will be no antiport occurring in this. So here the calcium comes to accumulate inside the myocardial cell, making the myocardium to increase the force of contraction. Hence the mechanism of digitalis. Digitalis increases the force of contraction. Yes. Thank you guys. I hope you understand. Please share your feedback. I compiled in one page, so you can see a little bit congestion. Sorry for that. Thank you guys.